So welcome everybody. I wanted to begin with a land acknowledgement today that the University of Alberta acknowledges we're located on Treaty 6 territory and respects the history, language, cultures of First Nation, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. And I think as healthcare professionals, this is an especially important consideration as we practice every day. So I wanted to also thank you to Paladin for their support of this ATI seminar series. So without any further delay, I'll introduce our speaker for today, Dr. Stefan Tullius, who is Professor of Surgery at Harvard Medical School. He is the Joseph E. Murray, MD, Distinguished Chair in Transplant Surgery, Chief of Transplant Surgery, and Director of the Transplant Surgery Research Laboratory at the Brigham and Women's Hospital in Boston. He's the Vice President of the Transplantation Society, also known affectionately by us as TTS, and he served as the Senior Treasurer for TTS. He's the Executive Editor in Transplantation, Vice President of International Society of Uterus Transplantation, which is obviously a really interesting subject, and he leads productive NIH-funded research lab. He's an international speaker at many meetings, I guess including our seminar today. And he served and has served in many, many societies, such as the European Society of Transplantation, TTS as mentioned, the United Network for Organ Sharing, or UNOS, and the National Kidney Registry in the United States. So he's also co-chairing several committees and basic science committees for several groups. And today he's going to talk to us about aging and alloimmunity. So thank you so much for joining us, Stefan, and I will turn over to you.
Thank you very much. And thank you very much for the very nice introduction, and very happy to be speaking to you today. I'm going to talk about aging and alloimmunity, which I think is going to be a topic that is of interest now and is going to be of many more relevance in moving forward. I want to now, let me see. So I have had trouble moving this forward. I want to start off by showing you some of the relevance of the aging population that we currently see. This is the rate of elderly dependence ratio, and what you're looking at is the frequency and the expected contribution of elderly in the overall population. And you see here, as the color is turning more darkly green, the higher will be those who are older as a ratio of the overall population. And you see that we currently have and will even see more an increasing aging population. And that is right now particularly relevant in North America and in Europe, but it will be of relevance throughout the entire world. Now, older patients do not only have a significant role in our overall society. You see also that the patients that we transplant currently are getting older. The green line here shows you that the overall rising population contributing to our overall transplant frequency are those over the age of 65. So more and more older recipients are getting organ transplants, and it is consequently also relevant that we will see and that we do have more and more older donors. So it's the older recipient that we will need to determine if that older recipient is going to improve and benefit from a transplant. But then it's also the older organ where we need to define how to utilize an older organ in the best way, that we need to have ways that we can assess the organ quality of older organs, and that we can try to understand if an older organ can be improved in its quality, that the older organ is going to cause a specific alloimmune response, which is different than that of a younger organ. And overall, it will be of relevance as more and more older organs are available that we utilize those in the best way. The dilemma of supply and demand is one of the most pressing issues in organ transplantation. Now, this here is from a publication that came out in the New England Journal in 2018, where we reviewed opportunities on how we can best utilize marginal organs for transplantation. But what you see here, and most relevant, is the discrepancy between demand and supply. We have now in North America, in the United States, increased our overall deceased donor volume. However, the discrepancy between demand and supply still continues, and wait times are excessively long in this country as they are in Canada as well. Now, what can we do? And to start out with the effect of aging on the organ and what we can do to utilize older organs in the best way, what can we do overall to increase and meet the opportunities for the demand of organ transplantation? I'm just listing here the different ways, the theoretical ways that we can approach this: xenotransplantation, regenerative medicine, increased living donor organ transplants, and here particular paired kidney exchange has been playing a significant role. But overall, utilizing available human organs is a realistic way on how to meet the demand, and is a way that we have been utilized and worked on during the past years. And those are numbers that show you that there is really an opportunity of utilizing older organs. This is a UNOS study that shows particularly that in the age group above 50, that we have a significant amount of estimated potential donors, and that the amount of unrealized donors is particularly high in that age group. So there is great potential to utilize older organs in the best way. And at the same time, we know that older organs come with shortcomings. And when we look into the physiology of an older organ, then we know that there are specific aspects that are characteristic of an older organ. There are fewer nephrons in the older kidney, for example. There is a decline in the renal function and a hypertrophy of the remaining nephrons. Fibrosis and glomerulosclerosis goes along with physiological aging. There is more of a sensitivity to inflammation in older organs, and we know from clinical data that older organs are more sensitive to prolonged ischemic times. And there is an aspect that is particularly relevant, and we have focused on that, which is the accumulation of senescent cells, which we see in the kidney, as well as in other organs as well. And I'm going to show you data on this. And we have tried to interfere with the accumulation of senescent cells in older organs and utilized that approach to improve the quality, reduce the immunogenicity, and reduce the sensitivity to unspecific injuries. Going back to experimental models in which we tried to study the way that older organs promote immune responses. Here we have looked into experimental models, mostly in mouse, heart, skin transplant models. And we have established a few years back that the transplant outcome of a young donor compared to an older donor, that the young donor organ survives longer, and that the immune response and the rejection of an older organ occurs earlier. This is then confirmed by histological analysis that we did, rejection scores, and infiltrating relevant immune cells, CD4 and CD8 cells, which you see down here on this bar graph. So more immune cells, a more potent immune response to older organs that is then relevant and relates to earlier rejection rates. And that was, we thought, was an interesting observation that we wanted to understand in a better way to see what the mechanisms are behind that. And one of the first questions we asked ourselves, if we see a more potent immune response to an older organ, is it the older organ per se, the parenchyma, that is causing that, or is it the cells that are in the older organ, that are transplanted with the older organ, and particularly dendritic cells that make the difference? And to get closer to answer these questions, we did create chimeric animals. We reconstituted a young donor with young bone marrow cells, or an old donor with young bone marrow cells. So we had either a young or an old kidney that came with young bone marrow cells. And if we do this, if we transplant those organs, then we do see that there is no difference in regard to the survival of those organs. So it seems that the graft passenger leukocytes, dendritic cells, are playing the most relevant role when we look into the immunogenicity that is related to the transplantation of older organs. And again, here confirming data in regard to histology and the rejection score that we did in this study. So on the flip side, then, if we think that dendritic cells are very relevant to be causing this augmented immunogenicity, we looked into ways of depleting dendritic cells, and that can be done chemically with the application of clotronate, which targets macrophages and dendritic cells. And again, if we depleted those cells from old organs, graft survival was comparable, and the histology and the rejection score was comparable, and then there was no difference in regard to an organ age-specific alloimmune response. Now, we asked ourselves, what is the driving factor? What do those dendritic cells cause in the recipient in regard to an augmented immune response? And in analyzing that, we found that the immune response elicited through the old dendritic cells is an interleukin-17-driven alloimmune response in recipient animals. And we could see this and confirm this if we either then treated the recipient with an anti-interleukin-17 antibody, or if we did transplants in interleukin-17 knockout animals. In those cases, there was no difference between the transplantation of an old and a young organ. So in concluding from those experiments, we saw that older organs elicit a more potent immunogenicity that is communicated through dendritic cells that cause an interleukin-17-driven, more potent alloimmune response. We then we continued those studies, and this here is a more recent study that we did in mouse heart and skin transplants as well, where we asked ourselves, so what is then causing the activation? What makes dendritic cells in an older organ more immunogenic compared to dendritic cells in a younger organ. Now, what you see if you compare old and young organs, skin, heart, and kidney here, is that there are more senescent cells accumulating in older organs. Senescent cells are characterized by specific markers. One of the challenges in the field is that there is not a single marker for the dendritic cells, but that the expression of several relevant markers, PT21, P16, is then characterizing senescent cells. And the characteristic aspect of senescent cells is that they are apoptotic resistant and that they spread a senescence-associated inflammatory product, which is then causing inflammation and causing the accumulation of more senescent cells. So you see here, with increasing age, more senescent cells. And this is true for skin, heart, and kidney transplants. And what we also see is when we have more senescent cells, that we see an accumulation of cell-free mitochondrial DNA. And then we try to understand what is the consequence, what is the accumulation of senescent cells with the accumulation of the mitochondrial DNA then doing to dendritic cells. And we wanted to see in this study that this effect is not only relevant for an animal model, but is also relevant when we look into the comparison of the accumulation of senescent cells and the accumulation of mitochondrial DNA in older donor organs. And what you see here is comparing organs from younger donors, here kidneys, and older donors, that we see an accumulation of mitochondrial DNA also in human samples. And that accumulation of mitochondrial DNA then goes in parallel with an activation of dendritic cells. So the observation that senescent cells accumulate, that senescent cells are the cause and the producers of mitochondrial DNA, which is then activating dendritic cells, is an observation that we saw both in experimental and clinical samples. Now, going back to the clinical, to the experimental approach, we were asking if we then deplete senescent cells in our mouse transplant model with a cocktail of senolytics, then would we then be able to reduce the accumulation of senescent cells? And would we be able to reduce the accumulation of mitochondrial DNA, then resulting into a prolongation of transplant outcomes? And what you see here is that the application of senolytics is causing the reduction of senescent cells, is causing a reduction of mitochondrial DNA. And if we take a transplant, a heart transplant, from a mouse that has been treated with senolytics, then brought into a system with an immunosuppression of using CTLA-4 Ig, then you see here on the orange line that the old heart that has been treated with senolytics is doing as good or even a tiny bit better than the young heart that has been treated with PBS as a control only. So the application of senolytics to a donor reduces senescent cells, reduces the amount of mitochondrial DNA, reduces the activation of dendritic cells, improves the quality, and I'm not showing you those data, but we did studies as well where we looked into models of ischemia reperfusion injury, where senolytics could significantly contribute to an improved quality and recovery post ischemia reperfusion injury. And with that, we are currently trying to move those experimental observations into a larger animal models and then hopefully to a clinical application. And as you know, organ preservation has really made great strides. And this is here showing Volker Belzer with one of the very early organ preservation devices. They are now way more sophisticated. Organ preservation devices are currently used at hypothermic conditions, at normothermic conditions for lungs and for livers. And it is possible that we can improve the quality and the immune response, reduce the immunogenicity of older organs, and that can be done in theory through an application, through a treatment of the donor, through the allograft treatment itself, and maybe also short-term after transplantation. So this is what we have and what we have been working on in regard to the aspects of donor age. Now, in addition to aspects of aging on the organ, there is an obviously very broad consequence of immunosenescence in regard to the aging immune response. And I'm just mentioning a few aspects that are important here. Clinically, we see when we transplant older recipients that there is an increase in the rate of infections, more infectious complications, higher rates of malignancies. We see a broad effect on various T lymphocyte populations. We see a decline of the naive T cell pool, an increase of the memory T cell pool, and significant changes to the humoral immune response as well. I'm just showing you a few more general aspects on how aging is impacting the alloimmune response, and will then show you what the effect of immunosuppressants in aging is, and that there is an age-specific aspects of immunosuppressants as well. Now, starting out, when we transplant older recipients, one of the first steps we need to do is we need to assess, does an older recipient really benefit from a transplant? And here it is very important to look into aspects of healthy aging. Those who are chronologically old, but remain biologically fit, are candidates that will certainly benefit from a transplant. And at the same time, those who have already signs of frailty will benefit less from a transplant and may not be considered. And that is very important to evaluate patients very carefully to make that distinction. Frailty and transplantation is a very important aspect. Frailty by itself is fairly poorly understood. There are several factors that play a role that result in frailty: chronic inflammation, metabolic changes, inactivity, malnutrition, caused by several aspects. Physiological aging is playing an obvious role. Genetic composition, comorbidities, and the environment. And it results then in a higher rate of mortality and in reduced and compromised post-transplant outcomes. There are several ways that we can assess frailty. There are those clinical established criteria, the free criteria. There's a frailty score. Those frailty aspects have been implemented and studied in clinical studies in and outside of transplantation. And there are obviously more detailed way of analyzing aging. The biological clock assessment is one way. Demethylation processes is another way that we can assess aging in a way to determine if a patient will benefit from a certain procedure. Now, I already mentioned aspects that are important in regard to immunosenescence and alloimmunity, the reduction of naive T cells, the increase of terminally differentiated memory T cells, and the presence of more virtual memory T cells that we see with aging. So important to understand that the ways of antigen presentation, TCR signaling, proliferation change significantly between the response, the alloimmune response of a proliferating naive and a senescent naive T cell, and that is playing a role in all aspects of the alloimmunity and our way of competently responding to pathogens. Memory T cells play a critical role. They are more frequent in immunosenescence, yet their capacity to respond in an efficient way is compromised. They produce less interleukin-2. They take on aspects of innate immune cells, and the response of memory T cells becomes unspecific. And likewise is the interplay between senescent cells and aging of other organ cells impacted. We already talked about the activation of T cells through mitochondria, the potential of interfering with this process through the application of senolytics. Then the production of the senescence-associated inflammatory product, which is represented by several critical inflammatory cytokines, and that effect then has an effect then on stroma cells and the aging of stroma cells, and then creating a vicious cycle where senescence in other organs is promoted through the senescence, through the senescent immune response as well. Now, one thing is particularly interesting, which is that we look into the adaptive, when we look into the adaptive immune response, that is with aging and with the chronic antigen exposure, very much like the chronic alloantigen exposure, turning from a specific adaptive immune response to an immune response that has more features of the innate immunity, and some aspects that are compromised in the adaptive immune response in aging are taken over through a less specific and more innate-like immune response. I want to show you a few data that will make it clear that the response to immunosuppressants in aging is very different, and that immunosuppressants, some of them act in a more effective way, but some of them do not and seem to be less effective in aging. We looked into tacrolimus, and we studied the effect of tacrolimus again here in a mouse transplant model, where we either applied doses that were comparable in regard to the milligram per kilogram dosage, and looked into the effect of transplant outcomes in old compared to young recipients. Now, in old recipients, graft survival is usually longer, even if one is not using immunosuppressants. If one is using immunosuppressants, in most cases, one can prolong graft survival even furthermore. So you see here that is a significant difference in regard to the outcome of in this experimental model on graft survival if we applied tacrolimus in the same dose in either old or young recipients. If we use the tacrolimus dose in a weight-adapted way, then we still see that there is a significant improvement of graft survival in older recipients. So in this study, we asked ourselves, number one, what are drivers for this observation? And is this, again, relevant for a clinical situation as well? We analyzed different immune cell compartments and immune responses. So we saw that the interleukin-2 response is specifically suppressed with the application of tacrolimus in older recipients. And we also saw that the proliferation of the CD4 T cell population is particularly specific and targeted by tacrolimus in older recipients. Now, there is a specific aspect that helped us to explain why tacrolimus is more effective in aging. If we looked into the concentration of calcineurin that was specifically reduced in older CD4 cells, and the effect of calcineurin expression and calcium influx was reduced both in older recipient in an experimental as well as in a clinical study. And that may explain and help us to understand why tacrolimus is more effective in older recipients. We looked into rapamycin, and rapamycin, one would think, is one of the agents that comes to mind early on to be used when we transplant older recipients. Rapamycin indeed prolonged graft survival in older recipients specifically. And the interesting aspect was that this was related to a specific population that was interferon gamma positive, interleukin-10 positive, a T regulatory population that was positive for those markers, which we saw subsequent to the treatment of older recipients and did not observe when we treated younger recipients. Again, this is experimental data utilizing rapamycin. Now, you may say that the immunosenescence is an overall compromised immune response, a less effective immune response, and therefore the way to treat older recipients is just to give them less of the same immunosuppression. As I've shown you before with tacrolimus and rapamycin, that is not exactly the case. Indeed, when we treated animals with a costimulatory inhibitor, CTLA-4 Ig, then we saw that this costimulatory inhibition was particularly effective in young recipients. So young recipients that were treated with CTLA-4 Ig in this experimental model had a graft survival of 100% in this particular strain combination. Old recipients treated with the same amount of CTLA-4 Ig and the body weight adapted amount of CTLA-4 Ig had a significant inferior graft survival. So asking ourselves again why we would see an age-specific difference. What we do see when we look into the expression of CD28 as the target for this costimulatory inhibitor, then we saw that in antigen competent and exposed cells, that the amount of CD28 negative cells was significantly increased in older recipients, and helping us to explain that CTLA-4 Ig is less competent and is less effective in older recipients. And indeed, if we then did an experiment where we transfected old cells with CD28, then we saw that if there was a transfection and CD28 expressed on old CD4 cells, that then the CTLA-4 Ig became as competent in older recipients. Another way, and that may be more innovative, to approach the aspect of immunosenescence in aging would be through specifically targeting metabolic processes. Metabolic processes are different in immune cells of old and young recipients. And we did here a study, an experimental study, where we treated old recipients specifically with an agent that prevented and interfered with glycolysis. Then we did see that there was an increased and specifically prolongation of graft survival in older recipients. So that may be another way of dealing with the specific aspects of immunosenescence if one targets metabolic processes that are specific for aging. Now, one aspect that you may ask yourself, and that we have asked ourselves, is if aging is so relevant, and if we transplant an old organ or a young organ into an age-discrepant recipient, is it possible that we could either enhance rejuvenation or accelerate aging? And what you see here, when we transplant an organ that has a different age than the recipient, then donor cells may immigrate to the graft. They then may transfer certain factors that will either contribute to an accelerated aging or a rejuvenation. So at least in theory, this seems to be a potential that we could observe after transplantation. We have looked into this also in experimental model, and what we did here was we transplanted either in a combination of young to young hearts from 3 months old mice into 3 months old recipients, or we used old hearts and transplanted them in 3 months old recipients. And what you see here is that with the transplantation of a young recipient that receives an old heart, we see an accumulation of senescent cells in organs outside of the transplant, lymph nodes, liver, adipose tissue. And we were then asking ourselves, is that just a bystander effect, or will we see an effect also when we are going to analyze the physiological readout in regard to physiological capacity? And I'm going to show you that in a second in regard to cognitive capacities. And what you can do is you have mice running on a running belt, a treadmill. And what we did see here is that the grip strength of mice that received an old, of young mice that received an old heart was reduced. And that we saw that the running capacity was reduced in recipients of old hearts as well, compared to the recipients of young hearts. Now, this is early studies. We looked into this at an outcome after 1 to 2 months after transplantation. We compared this to sham controls. And during that time, early on, we were able to see an effect on the physical capacity of those mice. We are currently looking into more long-term outcomes and effects as well. Now, another aspect that you would want to know about is, will the transplantation of an older organ into a young recipient also affect cognitive capacities? You can look into this with several ways. There are ways that you can put up a maze, a novelty maze, where you have this triangular shaped construct here. You pluck one arm of those. The mice are used to run around in this area here. Then you open this arm and you measure the time, how long it takes them to get into this novel arm and how much time they spend in there. So to see what the degree of trying to explore this novel arm in the mice will be. There's another way that we can look into this, which is called a water T maze. And this is this structure here, where you see that there is on one side an escape platform. So the mouse starts to swim, and then the mouse gets to know that here is a platform where the mouse can rest. So it gets used to this environment, and then you change the platform, and then the mouse is going to swim around and is going to go over here, and you measure the time it takes until the mouse is going to find the new escape platform. What we did see was that the transplantation of an older organ increased the amount of time that was spent in the novelty arm, and it had a comparable time was spent once the novelty arm was detected. But it took those mice significantly longer to find this, and then they spent less time in the novelty arm. Likewise, mice took a longer time that received an older transplant to find the escape platform once it was moved. So again, in those early studies, we saw that there was an effect on the transplantation of an older organ into a younger mouse in regard to cognitive capacities. And then we asked ourselves, finally, can cell analytics change and prevent this transfer of aging? And indeed, they can. If we treated animals, then they spent and had a longer, spent a longer time and ran faster in the rotarod test. And when we treated them, they again had less mitochondrial DNA accumulated. So in summary, older organs elicit a more potent alloimmune response. We have shown this in our experimental studies. And we have shown the effects also in human studies, where we see an accumulation of more activated dendritic cells. The immunogenicity and the outcomes of older organs can be improved when we deplete senescent cells. Immunosenescence on the recipient side has broad consequences on alloimmunity and transplant outcomes. Important to understand that established immunosuppressants have age-specific effects, and it is not just that the immunosenescence process is an immune response of a less robust, representing a less robust response. It is indeed an age-specific immune response that we need to understand better. Immunosuppressants therefore need to be tailored in older recipients, and in early studies we have shown that aging can be transferred in experimental models. With that, I wanted to thank you for your attention, thanking those who have over the years contributed to all our experimental studies, clinical studies, members of our lab, our support here, so the NIH, through our Kidney Health Research Fund, collaborators, Jim Kirkland and Tamara Chkonia from the Mayo Clinic. Dr. Kirkland and Tamara are the pioneers in developing Senolytics. My clinical partner, Dr. Milford, one of my clinical partners with whom I have done some of our clinical studies and our transplant laboratory manager, Hao Chow, who has been instrumental in getting all those studies on the way. Thank you very much again.
Thank you so much for such a thoughtful talk. Lots to consider. And again, I apologize, my video is still not working, so you just have to stare at a picture of me and hear my voice. We do have one question in the chat, but and I had posted a note also that we can, if you want to unmute and go ahead. I see Dr. Halloran already, Phil Halloran, I guess I should clarify, we have Halloran squared here. So Dr. Halloran, if you want to go ahead and unmute and ask your question, and then we'll go to the chat questions.
Stefan, it's good to see you, and congratulations on a fine body of work. I've followed this over the years.
Thank you.
I think it's this analytics really has potential. We've got another window on this through the MMGX system. We've got something like 5,000 kidneys and hearts. And so our window, first of all, we did study aging in mice. Aging in mice and aging in humans is sort of a different event. Of course, mice have telomeres about 4 times longer than yours, probably 8 times longer than mine. But I think the idea is that they use certain mechanisms controlling cell cycling, like the P16-4A mechanism, more than humans do. We use both, but we use telomere shortening mechanisms to stabilize ourselves during aging, and mice don't use them at all. But so I always have been cautious about watching mouse experiments, as I'm sure you have. And of course, also, obviously, mice, very young mice, have no memory cells, and that makes them quite different from the young adult human. And so their immune responses are different. But so that's one aspect that I always keep in mind when I'm watching this very important work. The second is we've looked at our populations, and there's a terrible thing goes on in the human population. It is obligatory that we distribute organs that are young to the young. And it's a bias in the whole field, which we can't escape from. And you simply can't statistically remove the bias. And if a young person gets an old organ, that person is probably at higher risk because they're in a different situation, because that's the way we work. And so when we looked at donor age and recipient age, we see a kind of a pairing. And if you look at, and you're seeing sort of the same biology because we're pairing them. And it's extremely difficult to escape that in our analyses. But the third thing we see is that at the window we have is the biopsy, particularly the indication biopsy. And what we see is aging sets the stage for a much more profound injury response. We don't see it primarily as an immune problem so much as an injury and repair problem. And in 2 ways. One is there's more injury, and secondly, the injury is not dealt with properly in older tissue. So those are just some comments. One is, how do you think about most human differences? Secondly, how do you think about the pairing between donor and recipient age? And thirdly, don't you think that really the big problem in transplanting old tissue is the massive accelerated injury response?
That is all very relevant, and we are aware, and the mouse aging process certainly is very different than that what we see in humans. What we have done is we have done in a significant amount of our work then tried to translate this into a clinical relevant scenario, for example, using the human samples that we got from another collaborator, Klaus Niemann at UCSF, and looking into the upregulation of mitochondrial DNA in there, and the relevance of old dendritic cells and the accumulation of senescent cells. So yes, we understand that the mouse model is a model. It's guiding us to find specific leads, which we then try to confirm in human studies. The second argument, your second point of the transplantation. The transplantation is biased when we have major age differences. That is correct. We did a few years back with Edgar Milford a clinical study of the UNOS database, and we looked in a multifactorial analysis specifically on the effect of both recipient and donor age on the rates of acute rejections. And the overall rates of acute rejection is going down with increasing recipient age, and it's increasing with increasing donor age. What we did then in this study was that we formed a correlation of donor and recipient age. And when you look into a large database such as the UNOS database, then you find combinations also where an old organ is being transplanted into a young recipient and the young organ is being transplanted into an old recipient. And the combination, and again, you're right, we cannot exclude that there have been other factors that played a role there as well, but we try to address those still in a multifactorial analysis. Transplanting an older organ into a young recipient had an acute rejection rate of 40% during the first year. Transplanting a young organ into an old recipient had the lowest acute rejection rate. And the rejection rate when transplanting an older organ into an older recipient was still significantly higher than a combination of a young recipient and young organ into an old recipient. So the aging effect seems to play a role in this analysis and in a clinical study as well. And the other aspect that you mentioned is what is the relevance of injury in an old organ? I think that's very true. I didn't show you, we did studies where we looked into the effects of senescent cell accumulation and ischemia reperfusion injury, and then the effect or the impact of depleting senescent cells prior to the ischemia reperfusion injury. And senescent cells seem to play a significant role in that context as well. So yes, aware that there are experimental challenges, but whenever possible, we try to either correlate it to clinical studies or to clinical samples. I think, with the additional, getting you and your information linked to what we do would certainly provide us with a significant amount of additional information.
Thank you.
Sounds like an opportunity for collaboration if I ever heard one. I'm going to, there is a question in the chat, and Dr. Anderson, if you want, I can just read it out, but you're also welcome to unmute and just ask the question yourself. So I'll go ahead.
Okay, sure.
Oh, there you are.
Go
Great.
ahead.
Yeah.
Thanks for your talk. I was just not 100% sure if I got the chimeras you created correctly, but I'm interested in the increased mitochondrial products that stimulate DCs from coming from aged donors, are they derived from the donor hematopoietic cells or the organ parenchymal cells?
So when we created the chimeric model, then we transferred hematopoietic cells. So we isolated the dendritic cells from hematopoietic cells. We did not isolate them from the graft itself.
And they were young ones going into an old organ, is that correct?
Correct.
And so in that case, you see decreased rejection.
Correct.
So does that not imply then that the young dendritic cells are less responsive to mitochondrial DC stimulators, which would still be coming from the aged organ? And that's kind of surprising.
Say that again. The young dendritic cells.
Well, because the rejection is less, it sort of implies that the young dendritic cells are responding less to mitochondrial products of an aged donor than aged dendritic cells.
So when we transplanted the chimeric organ that contained either young or old bone marrow derived dendritic cells, then we did not see a difference in graft survival anymore.
So there's no difference between whether the hematopoietic cells you put in are young or old.
No, the background of the organ being young or old did not make the difference in regard to graft survival. It was the age of the dendritic cell that determined the increase in immunogenicity. The dendritic cell came from an old donor.
Okay.
Then you saw the increased immunogenicity. If it came from a young donor, then the immunogenicity was not increased, and was independent from the age of the parenchyma.
I see. So it's the hematopoietic cells that are generating the mitochondrial products.
Correct.
I see. Okay, thank you.
We're right at the time. I don't see another question in the chat, but I'm going to take moderator's prerogative and just ask a less technical question, hopefully not as complex to answer. I noticed that one of the things that we see often, I'm in the HLA lab, so we'll see patients being relisted for transplant later, sometimes after a very long history of immunosuppression. So now they're older, but they're not like a typical, say, 60-year-old. They're a 60-year-old who's been on immunosuppression for, say, decades of their life already. Do you have any studies or models that sort of address that sort of real scenario that we see often in transplantation, or what are your thoughts about that?
I think that's an excellent point. What we see, for example, is that disease processes per se accelerate aging. So, for example, end-stage renal disease increases aging. I would speculate that chronic immunosuppression does that as well, but we haven't studied that.
Okay, well, yeah, it's just something that we see often in our patients being relisted. And so I've often wondered, like, what does that mean? It must be an altered immune system, you would think. And, I know I don't know if we have patient partners on this particular call today, but definitely immunosuppression, as you described, tailoring immunosuppression, it would be a holy grail for patients who, obviously live with many consequences to the immunosuppressive protocols that they live with every day. So it's really exciting work that you're doing.
Thank you.
In the interest of time then, even though I'm sure we probably could talk about this a lot longer, we just want to thank you again, Dr. Tullius, for being with us today. It's happening on May 10th. And I really thank everyone for their attendance, as well as, of course, our speaker. Thank you so much.
Thank you.